I'd like to introduce our next speaker. I'd like to introduce Chris Bahl, who is the President and Chief Scientific Officer and also co-founder of AI Proteins, which is a Boston-based firm. He holds a PhD in biochemistry from Dartmouth School of in the School of Medicine there, and got a master's degree in biochemistry and a bachelor's degree in biochemistry, molecular and cellular biology from the University of Maine. Chris did his postdoctoral work in David Baker's lab at University of Washington, where he pioneered design of de novo mini proteins there. And before starting the AI Proteins company, he was the founding faculty member of the Institute for Protein Innovation in Boston, and he also holds co-appointments at Boston Children's Hospital and also Harvard Medical School. So we're happy to have him here to talk about mini proteins.
Thank you very much. Excited to be here today. I'm really enjoying the kind of like the pop star microphone that I get to use today. I think if this symposium was like a 2-day music festival, by the end of it, people are kind of tired. You've been rocking out hard, or maybe in our case, geeking out pretty hard for a couple days now. So I'll do my best to keep this exciting and fun. So we've also seen a lot of really stellar talks over the past couple of days about using AI to understand data, to make inferences, to make predictions, and to help us kind of move faster and do more. This talk will be a little bit different. I think we rely more on generative AI to create useful things. What we want to create is proteins that can do something that may be really difficult to engineer using approaches that are more traditional. And so we're designing proteins totally de novo. So from scratch, these molecules have no evolutionary history, no similarity to anything that has existed before. And there's a very good reason why we choose to do it this way. When you create a molecule or a protein from scratch, you have so much more control over the properties, the behaviors, the activity of these molecules than is possible to achieve by modifying a natural material. If you'll maybe permit a brief digression, if you think about the way that humanity has constructed buildings from sort of the dawn of civilization until pretty recently on a historical perspective, we made our buildings out of natural materials as well. But we didn't construct buildings like the one we're in now, or presumably the folks over in Germany are in now, using timber and stone. We couldn't just shape natural materials and fit them together to create these types of buildings. You needed more advanced materials where you have more control over their shape and their properties and their size. You need concrete and steel and glass. So creating proteins from scratch is sort of like that transition to concrete and steel. It's a major inflection point in technology, and also allows us to create molecules that have way more sophistication in their mechanism of action, and we can build things that are just not possible if you're starting with something that's lying around that you find in nature. Let's see, can we go to the next slide, please? Great. I always like to start off with an acknowledgement. So I'm very fortunate to have been able to launch this startup company. I was in academia before, and all of the postdoctoral fellows and research associates came with me to start this company. And actually, one of the really all-star administrators in the department came as well. So really fortunate for this whole team to come. And we've about tripled in size since we launched. And so this is the team as of last summer. So this is really their story. And I feel really it's a huge privilege for me to be able to be here with you today to share their story with you. So next slide, please. Okay, so what are we creating? What are we trying to make? We're using generative AI to design proteins. And when you can make a protein be what you want it to be, the kind of the question is, what do you want it to be? And with a technology like this, it's the potential not just to make incremental step forward, but to do something transformative. That's our goal. And so you see that little tagline at the top is very aspirational, right? The future of medicine. And apologies, I know I'm in the heart of crop science here, and not everything is about human therapeutics. The talk today will be largely protein focused. We'll show a couple of a brief vignette on actually creating medicine with it. But this technology has many applications outside of human medicine. So let's talk about mini proteins a little bit. So we can create them to be what we want, but what do we want? We want right around 45 amino acids in length. So at this size range for a polypeptide chain, you are correct to call it a peptide. But what makes mini proteins special and different from other folded proteins is that they spontaneously adopt a three-dimensional shape that we design in silico. And it's this three-dimensional shape that confers the remarkable properties to these molecules. So the shape is very rigid, and that rigidity confers durability. So you can survive acid and proteases, high temperature, organic solvents, freeze drying, all these kind of like horrible punishments that you would never inflict on a normal folded protein. We can do all this using just the boring, old-fashioned, canonical 20 amino acids. We don't require any exotic chemistry to achieve this, which is typically what cyclic peptides are leveraging in order to make them stable and bind tightly to their targets. So because we can use the normal amino acids, this means that we're genetically encodable. And because the structure is so strongly encoded into the primary sequence, anything that can stitch amino acids together can make a mini protein. So you can put them inside of things to genetically encode them in, like an organism. Any organism with a ribosome can make them. Or at 45 amino acids, we can still make these molecules on a peptide synthesizer, and you don't have to do anything exotic. So like any commodity, peptide synthesis group can make one of these things. And so that allows us still to access the full chemical biology toolkit. In the case where you might want to conjugate something to it, you just put a click chemistry handle there, get an azyme. One of the other, I think, really, not quite ready yet. Please go back. Thanks. One of the, I would say, other really cool things about mini proteins is that when we're doing de novo design with them, that is that we get to make them what we want them to be. And so there's a whole host of developability parameters that we incorporate upfront when we design these things. So oftentimes in like a traditional protein engineering workflow, you're creating your protein, you get it to work at all to do what it's supposed to do, and then you kind of figure out how to bootstrap all the useful properties that you need into it to turn it into a product. If it's a therapeutic, right, it needs to have a whole host of developability parameters. So one of them here for I know there are some protein geeks in the audience here that have a strong structural biology background. So I'm showing the electrostatic surface potential of this molecule here, and this actually is a crystal structure. So we know we can do this for real, and you can see it's reddish hue. And that means it's negatively charged. We know that positively charged macromolecules tend to be sticky to kidney and liver, and we don't want that. So we just design them to have exactly the charge that we want. And there's many other parameters like this that we can incorporate. Tryptophan and methionine tend to oxidize more readily. That's a problem if you want your molecule to be stable out in a field or in a human or sitting in a tube while it's waiting to go into a human. So we just don't use those amino acids at all because they cause problems, particularly in antibodies. So there's a whole host of these types of things that we can design in upfront so that as soon as the molecule has the activity that you want, it's ready to go. You don't have to then try and make it into a product. It starts off that way because you made it on purpose to be a product. Okay, let's go to the next slide, please. Okay, great. So one thing I'm not going to do today is go into details of the algorithms for how we create the mini proteins. And that's not because I'm not excited about it or don't want to share it with you. It's actually that we are in a blindingly fast renaissance of methods development going on right now. So probably by the time I finish the talk, the techniques that I would be telling you about would be obsolete. So instead, we'll talk a little bit about how we use this in a larger pipeline, and the individual algorithms can be substituted out when the next latest and greatest thing comes out. You can just kind of swap these components, but you'll get a high level of what you need and how to put it together. So always the first step with creating any of these molecules is first to model them in silico. So you're creating the structure, you're designing the sequence. And so this is what we're doing. We were doing this kind of back in the dark ages of maybe 3 years ago before AlphaFold was around, and you were using more physics-based methods like Rosetta to do this. So it was still possible, just much harder and slower. And so we're creating the structure and we're creating a binding site in particular, because that's primarily what we're doing with these. We haven't figured out how to turn small proteins into catalysts yet. So we're creating binding sites for typically other proteins. And there are two ways philosophically that we do our binder design. The first is the very intuitive way where you have your target you want to interact with, and you essentially wrap a molecule around that binding site. So the whole thing is really constructed to interact with that one specific site that we're trying to bind. So that works, and it works pretty well, but it's also pretty bespoke. It still takes a really talented protein designer who knows about how to use the software and the ins and outs to get it to work. It's not a black box you turn the crank quite yet. And I'll also argue that the vast majority of binder problems don't require that level of sophistication. So there's another approach that we use where it's sort of I would say a newfangled technique on an old-fashioned idea. And so this is library creation. So the second way we do it is we have pre-designed libraries. If you're familiar with the antibody world, you might call this a naive library if it was naturally derived. And so in this case, we've designed large libraries of proteins, but instead of designing those binding sites against a specific target, instead we design each of those binding sites to be maximally different from one another. So we have a tremendously large amount of diversity. So these are 10 to the 9 libraries, but instead now they're just covering a massive amount of different geometric diversities and chemical diversities. So there's many, many scaffolds in this. It's not one protein, one binding site that's changing. So there's just so much very high quality diversity. And then we can use more established techniques. Next slide, please, like yeast and phage display. And we can rapidly identify these binders. And kind of the dirty secret of the targeted design approach too is that it's pretty hard and it still fails most of the time for hard problems. And so in order to get a binder that works, you might need to test anywhere. If it's an easy problem, you could do it in maybe 100 different designs. If it's a hard problem, you might need 100,000 designs in order to get something that binds. And so we're still leveraging a lot of synthetic biology techniques like yeast and phage display to be able to rapidly screen through, regardless of whether we're using a kind of a naive pre-designed 10 to the 9 library or a more targeted, you want to bind to this particular site. So computation is great. Synthetic biology is great. But the next slide, please. You still have to make the molecule and see, and we can skip this for time, please. We still have to make the molecule and make sure that it does what it's supposed to do. So you need to express and purify and characterize each of these using good old-fashioned biophysics that you can accelerate with robots. So we've created some new methods. I think for the protein scientists in the room, I would love to geek out more about some of the unique ways that we engineered our some enzymes that allow us to accelerate the process and make it really easy. But we could produce up to 1,000 mini proteins every week, and then you do circular diachirism, SPR, make sure that it actually does what it's supposed to do. Most of the time, you need to optimize it. So then next slide, please. Actually, one more. We'll skip this as well. This one is really cool. So hopefully, I would love to talk with folks about this one later. So optimization is also a problem where it really kind of grinds to a halt for pretty much every modality out there today. And it's just a slow iterative process. But with mini proteins, one of the really cool things is that the molecule itself is short enough that you can get the whole gene encoded on a single oligonucleotide, and you can read through the whole gene on a long read on like a MiSeq or an XSeq. So the ability to just create massive synthetic libraries and then understand them using NGS is also, I think, really unique to the mini protein modality. So we just test every possible amino acid substitution at every position. And we also know that amino acid substitutions tend to interact with each other when you make them at the same time. Epistasis is normally thought about with like genes interacting, but that still happens within a same protein. And so it's also helpful to make combinatorial mutations and sample those. And so it's sort of a take on site saturation mutagenesis. We call it AWESOME. It's a false acronym. We're just being jerks. And so what this allows us to do, next slide, please, is to take really any molecule out of our initial screens that we express and purify. It might have modest activity here. You can see this is a micromolar binder by SPR, so nothing to write home about. The circular dichroism trace there for the protein aficionados, you'll see, we take it at room temperature, we cook it, and we bring it back down. And the very little difference between those traces essentially says that you boil this protein and it doesn't care. It's not unfolding. And so this is kind of the traits that we like, but it doesn't have the activity we like. So we put it through that awesome process. It can incorporate multiple mutations simultaneously. So the next slide, please. And typically in one round of this, it's fairly common. Sorry, the text is kind of running over there. It's pretty common to get 1,000-fold affinity improvement doing this one time. So now you can take any modest binder with nice activity and just bring it immediately to something that has the activity that you want. And importantly, this is also provides a rheostat knob as well. You don't always necessarily want to bind to oblivion. You might want something moderate depending on what your task is. Okay, let's go to the next slide, please. And so this whole process is really fast. And this is, I think, one of the exciting things when you combine artificial intelligence with synthetic biology and robotics together. It's not just buzzword bingo. Like when you can really do all these things tightly integrated, it actually is a massive accelerator. So the quickest we've gone through from concept to purified mini protein in a tube with nanomolar binding activity was 3 weeks. That happens maybe 1 in 5 times. And then when we need to optimize, that lengthens the timelines. And so then it's more like 12-ish weeks. And so this is really a lot of throughput. So the question is like, well, what do you do with all of this ridiculous discovery throughput? If you can make them this fast, then we can do many campaigns in parallel. We've done probably a little over 100 targets so far, in just the 2 years the little startup's been around. Next slide, please. So here's how we're thinking about it is trying to push through molecules ourselves into the clinic, as well as working with partners. And we would love to also identify opportunities outside of human health. We have limited capacity, so those are the types of things that we are going to rely on a partnership for us to be able to initiate non-therapeutic applications. But so far, we have a big chunk of oncology, big chunk in inflammation, and then metabolic disease and other. So the next slide, please. Okay, so let's talk a little bit about therapeutic applications. I'm running very shy on time, so maybe we'll skip ahead, please. So we'll talk about a vignette in oncology. So when you have a new modality, a lot of times people are thinking immediately just, well, you've got this newfangled thing. So what can you do now that was not previously possible at all using, the tools of yore? And I think one of the longstanding challenges in protein engineering writ large is multivalency. And, it's possible to do this with antibodies. You can get bispecifics. Sometimes you can get trispecific antibodies. I'd say those are often heroic efforts to create trispecific antibodies. There's a few folks that have made tetraspecific antibodies binding 4 things. But anything beyond that is sort of starting to get in kind of science fiction-y. These are natural proteins that didn't evolve to be in these weird formats, and you can Frankenstein them together, but they often get unstable and fall apart. The mini proteins don't have any evolutionary baggage. They're so well behaved on their own. They seem to have an infinite ability to be linked together like pearls on a string and create these big multivalent constructs. And we can tune the distance between each of these binding sites very carefully. We can change the order in which they appear, and we can change the number of times that they appear. All of these things are under our control. Simple genetic fusions allow us to do this. We can put intrinsically disordered amino acid sequences in between them, or rigid linkers, however we like. And so now, if you have a high throughput platform for creating individual functional domains and an infinite ability to just Lego them together to create molecules that are really sophisticated, there's all these wealth of applications that become possible that are very exciting. So then next slide, please. So one of the first ones that we decided to work on is in immune cell engagers. So just very, very brief review. You have an immune cell, you have a tumor cell. There's some proteins on the surface of the immune cell you might want to interact with. There's some proteins on the surface of the cancer cell that you might want to interact with. And you can build a bispecific or trispecific or heptaspecific, dodeca-specific protein that would link these together and target the tumor in a very sophisticated way by making sure it's interacting with multiple things on the surface, interacting with multiple different receptors on the surface of the immune cell. Maybe you're modulating cytokine pathways or checkpoint inhibition, all of that together and all of that activity localized right at the immune synapse. And so those are the types of things that become possible. Next slide, please. And so here's just one vignette of a very simple bispecific, but can go up to trivalent, just to show how rapidly we can do this. At the top, the little green mini protein there is the mini protein that binds to the tumor associated antigen on the surface of the, in this case, the AML cell. And then the little orange mini protein binds to the receptor on the immune cell and can help to activate it. And what you can see here is we're titrating the distance between these two binding sites. Could switch the orientation in the chain and then again titrate the distance. And then we can also modulate the valency and the linker compositions. And what we do is we mix cancer cells with immune cells, and then we do a flow cytometry assay to see how many of the tumor cells get killed by the cancer cells. And you can see right away, we get about single-digit nanomolar EC50, so efficacy in killing those tumor cells, which is really quite good. But with one round of optimizing the distance, the linkers, and the valency, we get to single-digit picomolar EC50 cell killing with near complete tumor cell killing. So now this is an incredibly potent immune cell engager, and it's still not that sophisticated. It's only 3 different binding sites. So I can see there's a red light blinking in the background. So let's go to the next slide. I'll wrap this very fast here. We did this in a xenograft mouse study, and it works. It controls human tumors engrafted into a mouse. And so that was quite exciting. Next slide, please. Okay, so here's the team one more time. Thanks so much, all of you, for your attention today. And yeah, I'm excited to take some questions.